right. Hello, everyone. All right. This is the re-recording of discussion one. I apologize for the time it has taken me to get this to y'all. The animations and the design of this PowerPoint have taken me way longer than expected, but I have finally pulled it all together and I am ready to go. So I'm going to give y'all kind of a preview of um, the spacing that I would do if I were doing this on the discussion worksheet, filling it along as well. So if you see, I've uploaded a new discussion one worksheet to the Canvas modules. And this is about how big my writing is for the first portion. So give yourself enough room, don't write too big. This is about how big I wrote for the first portion. But again, I'll be doing it over PowerPoint, so I won't be drawing along with you. So I just wanted to give you an idea of what we're looking at before we hop on in. So I'm going to go ahead and share my screen and we will start with discussion one, re-recording. So today we're looking at membrane movement and epithelial transport. So again, question one, what Dr. Chang is asking us to do is draw out a few different ions, molecules, and gases and talk about where they're in greater concentration and where they're in lesser concentration. So let's start off with our ions. So first we have, and real quick, just to give you an idea of the illustration here, the outside of this circle is representing the extracellular fluid or the blood, and the inside is representing the intracellular fluid, so the inside of the cell. So starting off with those ions, first we have sodium. So sodium is always going to be in higher concentrations outside of the cell and lower concentrations inside of the cell. So we have sodium, potassium, and calcium. Those are what Dr. Chang is asking us to label. And again, sodium is always, in almost every case, pretty much every case, going to be higher outside of the cell and in lower concentrations inside of the cell. Potassium is usually inverse or almost always inverse from sodium. So when we see sodium higher outside of the cell, potassium will be lower outside of the cell, meaning it's higher inside. Lastly, we have calcium. Calcium is normally in higher concentrations outside of the cell and lower concentrations inside of the cell. So this is important to know because one big thing with membrane movement is the use of concentration gradients. So if you remember back to basic biology, how ions and molecules move with their concentration gradient is when they go from high concentrations to low concentrations. So high to low. If they're moving against their concentration gradients, they're going from low to high, and that involves some energy. So going high to low involves no energy, low to high involves some energy. So that's the difference between passive and active transport. So now let's look at our gases. We have oxygen and carbon dioxide. And what we see is that oxygen is going to be in higher concentrations outside of the cell and lower concentrations inside of the cell. Opposing oxygen is carbon dioxide, and we see that it's lower outside of the cell and higher inside of the cell. And that would make sense because carbon dioxide is a byproduct of metabolism. So as our cells work and use energy and create products, the byproduct is carbon dioxide. So in our cells, we have the mitochondria, which are working hard to oxidize fats. We're working with different types of macronutrients and things like that, that whenever we use them, they produce carbon dioxide. So carbon dioxide, again, is higher inside of the cell because it's a byproduct of cellular metabolism. So the last molecule we're going to look at is glucose. So what we see is that glucose is in higher concentrations outside of the cell and lower concentrations inside of the cell. You might be wondering why this is, because if you're familiar at all with exercise physiology or cellular energy production, you know that glucose is what's broken down to form ATP for energy. However, one important thing to realize is that whenever glucose is inside of the cell, it undergoes a process called glycolysis. And glycolysis converts that glucose into glucose 6-phosphate. Glucose 6-phosphate is not the same thing as glucose. So we have higher concentrations of glucose 6-phosphate inside of the cell and lower than the outside of the cell because glucose 6-phosphate is the phosphorylized form of glucose, meaning that that glucose molecule is now locked in the cell. But again, glucose and glucose 6-phosphate are not the same. They're two different molecules. We have to undergo glycolysis to convert glucose into glucose 6-phosphate. So if you are wondering why we have lower concentrations of glucose inside of the cell and higher outside, that's kind of some reasoning behind that. All right, awesome. So that is our explanation and work through of question number 1. So let's move on to B and C. So these again are still referring to our membrane movement. So the first thing we're asked is which molecules can move by simple diffusion? The answer to this is our gases, oxygen and carbon dioxide. Water can also sometimes move by simple diffusion. However, we call that osmosis. So osmosis is a form of simple diffusion, but it is not simple diffusion. It deals with water specifically. Next, we have which requires some type of transporter. So whenever we talk about transporter, we're usually talking about a channel or carrier. However, we have different types of transporters or different subunits of transporters. They'll always work through a channel or carrier. However, we can have more advanced transporters like symports and antiports. We'll get there in just a little bit. However, when looking at the molecules or ions or particles that use transporters, what we see are ions and glucose molecules. Water can move by channels too, because anytime you open a channel for an ion to move through, water will naturally move through as well. All right, so let's look more closely at those two transporters, the carriers and the channels. So carriers and channels are a type of facilitated diffusion. They don't necessarily require energy. However, they require some form of channel or carrier. So what we see is that channels, and sorry, let me reword what I just said there. They aren't dealing with active transport because we're still moving from high to low. However, we need to use channels and carriers for molecules that are too big or have some type of charge to pass that cell membrane on their own. So those molecules still may be moving from high to low concentrations, which is a form of passive transport. However, facilitated diffusion is a form of passive transport. It just is there to assist those larger molecules or those polar molecules to move through the cells. And we'll see some cell membranes. We'll see some examples of this coming soon. So channels are membrane proteins that create holes in the membrane for ions to move through. So for example, sodium channels. So here's our cell membrane and here's a channel. It opens up and sodium can move from high to low concentrations. It's pretty simple. Carriers are membrane proteins that only open to one side of the cell for a molecule to move through. So here we have, you can think of glucose. Glucose is on one side of the cell. Glucose is a polar molecule, so it can't just diffuse simply past that membrane. We need some type of carrier to move it through. So what will happen is glucose will saturate this carrier. Once saturated, that carrier will open to the other side of the cell and glucose will be able to pass through. So that's important to remember. Carriers can be saturated compared to channels. Channels open and as many molecules or ions that want to move through can, compared to carriers. There has to be a specific amount of molecules to bind to that carrier to get it to open to the other side. And vice versa, we cannot bind multiple molecules to that carrier. It can only accept so many. There are only so many seats on the carrier to carry those molecules to the other side. So that's what we're referring to when we talk about saturation of carriers. So moving on, now we're going to look at our three main types of transport. We have simple diffusion, facilitated diffusion, and active transport. So I've kind of reviewed the first two. The last one we haven't gotten into much. But again, simple diffusion is working with molecules that are moving from high to low concentrations. So it is passive, it uses nothing, no energy, and no ATP. So what we see, what molecules or particles we see move with simple diffusion are small nonpolar molecules and also steroid hormones, which we'll get more into in lecture and in the coming discussions. Facilitated fusion is also the movement of molecules from high to low concentrations. However, we need some type of protein channel or carrier because we're working with large or polar molecules or ions that are charged. And we'll look at what ions are in just a little bit. Lastly, we have active transport. Active transport is talking about the movement from low to high. So we need some type of energy here because we're moving against our concentration gradient, that low concentration, the high concentration. We have two types of active transport. We have primary active transport and secondary active transport. We'll see some examples of this coming soon, but for now, just know that primary active transport uses ATP and secondary active transport uses ATP and some type of carrier. The molecules that we see that use active transport are any molecules that need to move against their concentration gradient. So any molecule that needs to go from low to high concentrations. And we'll see an example coming soon. So now let's go over those particles. Particles is the correct word. Molecules is very commonly used incorrectly. I've used it many times incorrectly in here. When we say particles, we're referring to ions, polar molecules, or nonpolar molecules. Molecules is normally the word most commonly used, but particles is what we're truly talking about when we're talking about all three. So specifically, our first particle is an ion. Ions are either positively or negatively charged and move via facilitated fusion or active transport. So if an ion such as sodium, chloride, potassium, or calcium need to move through that cell membrane, they need some type of carrier or channel to move through. And that is specifically due to them being charged. So if some type of particle has a positive or negative charge on it, that means that we're going to need that facilitated diffusion or active transport in the cases where we need to move it against its concentration gradient to transport that particle. Then we have our polar molecules. So a polar molecule is a molecule which one end is slightly positive and the other is slightly negative, and it will also move via facilitated diffusion or active transport. However, the example here is a little different. We see glucose. So glucose is a polar molecule. It is not an ion. It is a molecule, specifically a polar molecule. You'll definitely want to know that. And lastly, we have our nonpolar molecules, and this is one that has no net electrical charge. So it can simply move with simple diffusion. So the examples here are going to be oxygen, carbon dioxide, and nitrogen are gases. They can move via simple diffusion because they have no net charge. All right, moving on. So now we are going to get into that first chart under number 2. Sadly, PowerPoint would not let me animate this chart. It all just came out as one. So here are the answers to filling in this chart, but we're going to work through the reasoning behind why. So column 1 is looking at the movement of a specific type of particle into some area. Column 2 is looking at how it moves, and column 3 is asking why. So starting off with the first one, we have sodium moving from the extracellular fluid into cells. So again, we know that sodium is always in higher concentrations outside of the cell. So sodium moving from the extracellular fluid, the outside of the cell, into the cell is a form of passive transport because we're moving it from high to low. However, we don't use simple diffusion because sodium is charged. You can see that plus sign right there. So it's charged. Therefore, we're going to need to use facilitated diffusion to do so. And with our ions, we're normally just going to use a channel. So moving sodium from the extracellular fluid into the cell, we will use a channel, specifically a sodium channel. So why we do this is because, again, sodium is a charged ion, and charged ions need some type of opening in the cell membrane. They can't simply diffuse past. We also see that it's following its electrochemical gradient, and this is another one of the answers, kind of the main answer that you need to know. And we won't go too far into this. You may not even see it on the exams, but I did want to clarify it just in case. What we're talking about when we say following the electrochemical gradient is we're talking about how sodium ions are positive. And if you remember back to basic biology, like charges repel like charges. So positive repels positive, positive and negative attract, positive and positive repel. So that electrochemical gradient is talking about the repulsion provided by those positive sodium ions kind of pushing each other through that channel. They don't just kind of start moving, they are repelled due to that electrochemical gradient. All right, next we have aldosterone moving into a target cell. So just to let you know, aldosterone is a steroid hormone, which is small and non-polar. So what we see with our steroid hormones or our small non-polar molecules is that they can move via simple diffusion, via passive transport. There's no energy required to move those steroid hormones. As long as there's no protein attached to that steroid hormone, it will simply move past that cell membrane. As we get further into lecture, we'll learn about the tendencies for hydrophilic, hydrophobic, and lipophilic, lipophobic preferences with proteins versus hormones. And we'll get into that in discussion 3 for some further explanation. But for now, just know that aldosterone is this small non-polar steroid hormone, and those can move via simple diffusion. Lastly, or next, we have glucose moving into a renal proximal tubule cell and glucose moving into a muscle cell. And I'm actually going to use a diagram to explain this, and we're going to go pretty in depth with these because you do need to know how this works, and just simply giving the answer here is not enough. We need to know about the specific transporters that help us move glucose into a epithelial cell and then further move it into a muscle cell. So here are the answers. You can go ahead and write these down. And now we're going to move into the explanation behind why these are the answers. So here is a diagram that I drew out here, and I also included it on the updated discussion 1 worksheet. So this is what it looks like. So this is how I spaced out mine. It did get pretty condensed in there, so make sure you write pretty small. But this is how I would take notes on this portion of the discussion worksheet. And of course, you can always draw this out. You can always draw out what I have on the PowerPoint on a separate piece of paper if you would prefer. If you like to write really big, that might be the better option. So what we're looking at here is the lumen of the kidney, which whenever we're talking about the lumen, Dr. Chang likes to refer to it as the outside of the body. So it has not been absorbed by the body. It's in one of the tubes of the body, and we have not yet absorbed it. So lumen kind of is referred to as outside of the body in Dr. Chang's class. So that's what we have here. Then we have that renal proximal tubule cell that we were talking about in that chart. For now, just know of it as an epithelial cell. In lecture, you'll learn more about the details of the renal proximal tubule cells. I think that comes in unit 3. But for now, just know that renal proximal tubule cell is a epithelial cell. Then we have the ECF, aka the blood. And then lastly, we have a muscle cell. So what we're going to do here is first we're going to draw out our concentration gradients. So everything I'm writing in orange is in relation to this epithelial cell. So everything in orange is in relation to the epithelial cell. Once we get to the ECF, we're going to be talking about relation to the muscle cell. However, we're not there yet. So I would color code this for you. So again, we're going to first draw out our concentration gradient. So one thing that we hopefully have memorized by now is that sodium is always in higher concentrations outside of the cell, lower concentrations inside of the cell. Once more, higher concentration outside of the cell. Now, if you remember back to the first problem that we saw that glucose is normally higher outside of the cell. However, there is one unique situation, and that's with epithelial cells. Unique to epithelial cells, we see that glucose is in higher concentrations inside of the cell. This is the only situation where glucose is going to be higher concentrations inside of the cell. In all other cells, like our muscle cells, our liver cells, etc., glucose will be in lower concentrations inside. However, with our epithelial cells, we see that glucose is in higher concentrations inside of the cell. If it's in higher concentration inside of the cell, that means it's going to be in lower concentrations outside of the cell. So now we have our concentration gradients drawn out, and now we're going to work through labeling the transporters to get our sodium and our glucose into our muscle cell, because again, we're looking at glucose moving into the renal proximal tubule cell and glucose moving into the muscle cell. And what we see is that glucose needs the assistance of sodium to do so. So we're going to use 4 different types of transporters here. We have our sodium channel, our sodium glucose symport, our sodium potassium ATPase, and our GLUT carrier. So let's start working through these one by one to discuss and understand how each of them works. So first we have our sodium channel. And sodium channel is definitely going to be the easiest of all 4 of these. So a sodium channel, again, is a form of facilitated diffusion. So what we see is that we are moving with our concentration gradient from area of high to low concentration. However, we need that channel because sodium is a charged ion. So charged ions need some type of channel to move through the cell membrane, even though we're going from high to low using passive transport. So that's our sodium channel. Again, it is a form of facilitated diffusion, but because it's charged, we need that channel. Next, we have our sodium glucose symport. So sodium glucose symport. So what we see is that whenever we want to move glucose against its concentration gradient, so from an area of low to high concentrations, we will use some type of form of active transport. And we have 2 types of active transport, and the first is a symport. So glucose moves against its concentration gradient, and how it does that is by getting energy from sodium moving with its concentration gradient. So whenever a molecule moves from high concentrations to low concentrations, it gives off energy. Whenever a molecule moves with its concentration gradient, it gives off ATP. And whenever it gives off that ATP, it can be used by another molecule, such as our polar glucose molecule, to move against its concentration gradient. So that's why we call it secondary active transport. It is getting energy or ATP from a secondary source. Glucose is getting energy from sodium to move against its concentration gradient. And it's a symport because we see that those particles are moving in the same direction. So when sodium is moving into the cell, so is glucose. There's another type of secondary active transporter called an antiport. And we'll see an example of that coming up in just a little bit. And with antiports, they would be moving in opposite directions. And again, we'll see an example of that coming soon. So this is our sodium glucose symport. So we now have glucose inside of the epithelial cell. Now we're going to look at our sodium potassium ATPase. So this is some type of cell membrane carrier. So what we see is that we now need to move sodium into the extracellular fluid, but we need to move it from areas of low concentration to high concentration. So we need to move it against its concentration gradient. And anytime we need to move something against its concentration gradient, we need some form of active transport to do so. So here we're going to use primary active transport. Whenever sodium moves in one direction, potassium normally moves in the opposite direction. And when that happens, it gives off ATP. So the sodium potassium ATPase, that is some subunit that refers to potassium giving off energy when it moves in the opposing direction of sodium. It's kind of like the sodium potassium pump. Whenever we need to pump sodium in, potassium will be pumped in the opposite direction, giving us that ATP to move that sodium against its concentration gradient. So that is our sodium potassium ATPase transporter, a form of primary active transport. Sorry, this should say primary active transport. And it should not say antiport. This is primary active transport. So disregard the antiport. We're going to see an example of that on the next problem. This is primary active transport. So now we have the lumen of the kidney, again, the epithelial cell, the ECF, and blood. But now we're going to be talking about this in relation to the muscle cell. So all this time we've been talking about things in relation to the epithelial cell. Now we're talking about things in relation to the muscle cell. So again, sodium, there's no exceptions. It's always going to be in higher concentrations outside of the cell and lower concentrations inside of the cell. However, glucose in every situation other than epithelial cells will be higher outside of the cell and lower inside of the cell. So remember, glucose is always higher outside of the cell except in epithelial cells. So again, we're in the extracellular fluid, so we see that it's in higher concentrations than the inside of the muscle cell. Glucose 6-phosphate would be in higher concentrations inside of the muscle cell and lower concentrations outside. But again, glucose and glucose 6-phosphate are not the same thing. So now we're going to be looking at our GLUT carrier because again, our primary goal is to get glucose into the muscle cell. So how we use this is we simply open a carrier on that muscle cell membrane and we move glucose from an area of high concentration to low concentration. So we're moving with our concentration gradient. So it's a form of facilitated diffusion. It is a carrier, so it can be saturated. And this is all because glucose is a polar molecule. So even though we're using some form of passive transport, because we're moving from high to low, it is facilitated diffusion, which is still passive. We just need some type of opening of that cell to get that glucose through because it's a polar molecule. All right, awesome. So that gives us our answers to the last two columns of the chart. Hopefully you can figure out why those answers apply. It makes a lot of sense once you understand this process that we just worked through. Okay, great. So now we're moving on to our epithelial transport application problems. So just to give you, a review on what we're talking about when we're talking about epithelial cells or the epithelium, remember that the epithelial cells line organs. So they line the inside of organs. So we have the lumen of some type of organ, such as the kidney or the small intestine, and lining that is our epithelial cells. So we see our epithelial cells have an apical and a basolateral or basilar membrane, a basement membrane. So the apical membrane is the side that's closer to the lumen and sometimes called the luminal membrane. And then the basolateral side is the side that's touching the ECF or the extracellular fluid. And we're going to see some more examples of that coming soon. Here's just kind of another illustration. Here we have the lumen. This is specifically the lumen of the kidney because you can see excretion and filtration, which we'll learn a lot about that in Unit 3. But we have the lumen of the kidney, we have our epithelial cell, and then we have our ECF, the blood, so the blood vessels. So this is talking about some type of molecule moving in from the kidney into the ECF, and to do so, it has to pass through those epithelial cells, that epithelium, the lining of that lumen or the tube. So this first problem is talking about the transport of hydrogen and bicarbonate in the nephron of the kidney. So what we see is that the transport of hydrogen and bicarb play a major role in the acid-base balance of the body. Hydrogen and bicarb are synthesized or created within the epithelial cells from carbon dioxide and H2O in an enzymatically catalyzed reaction. I wouldn't worry too much about this or this. We'll learn more about that in Unit 3 when we get to the renal system. However, something important to note is that the movement of these ions, hydrogen and bicarb, are linked to sodium transport. So if you haven't put it together by now, sodium is a major component in transporting other molecules. So the relative concentrations in the net direction of movement are all shown in the table that we see on the worksheet, and we'll see it on the next slide. And we're being asked where should the specific transporters that we are given be placed, and what method of movement is occurring for the molecule. So here is that table with the different ions and what is going on with them. And then we have 4 different transporters that were given. I don't know if I wrote those out. I think I did them one by one in the coming slides, but we have the sodium channel, we have the sodium potassium ATPase, we have the sodium hydrogen co-transporter, and the sodium bicarb transporter. So how we're going to work through problems like this is we're going to first draw out our concentration gradients, and then we'll draw out our secretion and absorption goals. So sodium, as we know, is always higher outside of the cell, lower inside of the cell. Now, hydrogen and bicarb will normally be given. So what we see here is that hydrogen is high inside of the lumen. So that tells us that it is high inside the lumen, and that means it's going to be low everywhere else since it was specifically given. Bicarb, we were told that it's high in the cell. So if it's high in the cell, that means it's going to be low everywhere else because it was specifically stated. So we've now drawn out our concentration gradients. We can now see which molecules move from high to low or low to high. And now we need to draw out our secretion and absorption goals. So what I'm talking about here is this column of the chart. So whenever we absorb, we move from the lumen into the extracellular fluid. So we move from the lumen through the epithelial cell lining the lumen into the extracellular fluid. So what is stated that we are absorbing in this situation is if you can tell, I'm extremely tired right now. It's been a long day, but I promised y'all this video, so I'm gonna get it done. So what we are told is being absorbed is sodium and bicarb. So we can write that at the end of our arrow. Now we'll draw out our secretion goal. So secreting is going from the blood through the epithelial cell into the lumen to be secreted by the body. Specifically, when we're talking about the kidneys, we're talking about secreting it through the ureters, through the bladder, through the urethra, urinating it out. And the ion that we're given that is being secreted is hydrogen. So we're secreting hydrogen. So let's go ahead and work through how we can absorb sodium and bicarb and how we can secrete hydrogen. And how we're going to do so is with those four transporters given. So the first one we're working with is the sodium channel. And again, sodium channels are very basic and easy to understand. Sodium moves from high to low. So we open up those channels and sodium moves from high to low and high to low. However, we need to cancel one of these out because again, we have a goal with our sodium. We're trying to absorb our sodium. So when looking at our arrows, we see that this arrow goes in the opposing direction. This arrow goes in the secretion direction, getting it out of the body. However, our goal is to absorb sodium into the body. So therefore, we're going to cancel out this channel, because it is secreting sodium and we need to absorb sodium. So this is the channel that will remain. Next, we have our sodium potassium ATPase subunit. So this is some type of protein carrier as well. So again, we saw this in the previous question. We'll open up that channel or carrier, and what we see is that sodium needs to move against its concentration gradient anytime we're using some form of active transport, which sodium potassium ATPase is a form of active transport. We move against our concentration gradients. So we drew out our arrows moving against our concentration gradients, and we get energy from potassium moving in that opposing direction through that sodium potassium ATPase subunit. And so now we need to determine which one of these we want to keep to match our absorption goal. So again, we're trying to absorb sodium, not secrete it. So we would cancel this one out. So we have now used a sodium channel to absorb sodium into the epithelial cell. We have now absorbed it from the epithelial cell into the extracellular fluid, into the body. So we have now gotten our sodium inside of the body. So now let's go ahead and work with our sodium hydrogen co-transporter. So what we see here, and just a quick reminder, is co-transporters are a form of secondary active transporter. Anytime you see a co-transporter, it's a form of secondary active transport. And so let's determine whether it's a symport or an antiport. Those are our two types of secondary active transport. So we're going to open some type of channel here. And again, we're trying to move hydrogen outside of the body. So we're secreting hydrogen. So we draw out our secretion lines, and then we draw out our sodium lines. So what we see here is that sodium, if it's going to be a co-transporter, it's going to move in that opposite, or it's going to move from high to low or high to low. Sorry, I have my arrows messed up here. Let me draw them. This arrow should be pointing that way. Let me erase that other part. Let's see, pin. So if it's a co-transporter, we know that the other molecule that's not moving against its concentration gradient is going to be moving with its concentration gradient to get off that energy. So again, we're trying to secrete hydrogen from the body. So when matching this up with our goal, we can cancel out sorry, I am very tired. We can cancel out the basilar or the extracellular fluid side, because even though we secreted it from the body into the epithelial cell, that's still not removed from the body. We need to get it from that epithelial cell into the lumen. And so that's what we've done here. We removed it completely from the epithelial cell into the lumen so it can be secreted out of the body. Okay, awesome. So moving on to our last type of transporter and our last goal, we have our sodium bicarbonate co-transporter. One last thing to point out here that I forgot to mention is this is a antiport, because it's moving in opposite directions. So again, it's that form of secondary active transport, but it's an antiport because they're moving in opposite directions. So now we have our bicarb. Again, we're trying to absorb bicarb. So we open up that channel or carrier, and we draw out our arrows. So we know that it's a co-transporter, it's a form of active transport, so we're moving against our concentration gradients. So we draw that out, we're moving against our concentration gradients, so from low to high, low to high. And we can't do that without some form of energy from another molecule. So we use sodium because it's stated. So sodium will move with its concentration gradient. And again, our goal is to absorb bicarbonate. So whenever we work through this, what we see is that we will cancel out the luminal side because we are trying to absorb it into the body. So what we see here is that this is a form of, another mistake I made, I'm so sorry. This arrow should be pointing in this direction, and this should not be going here. It should be pointing in this direction. This is, again, a form of secondary active transport, but it is a symport because those molecules are moving in the same direction. And we're actually getting our ATP from the bicarbonate. So the bicarb is moving with its concentration gradient from high to low. So we're getting our energy from there. And that's honestly assisting the sodium to move from low to high. So make sure you work through these a few times and really get them drained into your brain. I would definitely work through these at least 3 times to kind of understand how these transporters work. I can explain it to you, but you really have to make it click in your head. All right, so here's our final cellular picture. So here we have that sodium channel, that sodium moving into the epithelial cell. Then we have that sodium potassium ATPase subunit, sodium moving into the body due to the energy given off from our potassium unit. So again, this is a form of primary active transport. Then we have our hydrogen being secreted from the body because that was our goal, was to secrete hydrogen. And we're doing so through that sodium hydrogen co-transporter, which is a form of secondary active transport, specifically a symport because they're moving in the same direction. And lastly, we have our sodium bicarb co-transporter. Again, anytime you see co-transporter, that means it's a form of secondary active transport. And this one is an symport as well. They're moving in the same direction. All right, awesome. So now that we have our chart drawn out, we can go ahead and work on answering these questions. So we can go ahead and work on answering these questions. So what we see is that in sodium absorbing epithelial tissue, such as tubule epithelium, the ATPase subunit will be on the ECF membrane, so the extracellular fluid side. So this is the ECF membrane, also known as the basilar or basolateral membrane. And that ATPase subunit is right here on the ECF membrane. Which membrane is the apical membrane? The apical membrane is the luminal membrane. So the luminal membrane is this one right here, and we can also know it as the apical membrane. So luminal and apical membrane are the same thing. Basolateral and extracellular fluid membrane are the same thing. On which membrane should you first find the, or on which membrane should you find the sodium hydrogen co-transporter? That's going to be, that's going to be the lateral, or the, luminal membrane, because right here we have that sodium hydrogen co-transporter, that symport. And then this transporter is an antiporter. Yeah, I had a feeling I made a mistake somewhere. Okay, let me pause this and figure this out. Okay, so I figured this out. I'm so sorry, guys. So again, what I was saying earlier is secondary active transport uses the energy of one molecule moving with its concentration gradient to move another molecule against its concentration gradient. Here you can see that I was saying that we are getting sodium, or we are getting energy from sodium moving against its concentration gradient, which is incorrect. So this arrow should look like this instead. Let's see, how can I follow this? Sodium should be moving inside of the cell and with its concentration gradient, giving off that energy to move hydrogen against its concentration gradient. So again, secondary active transport is moving one molecule with its concentration gradient from high to low. That gives off energy to another molecule to move against its concentration gradient from low to high. So that is a mistake that I made there. Go ahead and fix that on the previous slide. Hopefully you can fix that on your own. Again, sodium should be moving from high to low to give off that energy to hydrogen to move from low to high. So this is an antiport. They're moving in opposite directions. Where would you expect to find the sodium channel? That's also going to be the luminal side. This is our sodium channel right here. Given that the sodium bicarb cotransporter is on the extracellular fluid membrane, it is a symporter. So that's looking at this one right here, our sodium bicarb cotransporter, and they are moving in the same direction. So sodium is moving from an area of low concentration to an area of high concentration, and bicarb is moving from high concentration to low concentration. So the ATP is really being given off from this bicarb. I apologize when I put this into PowerPoint. I obviously mislooked my old notes. It's correct on my paper, but I made a mistake here. So we're actually giving energy to sodium to move into the body from bicarb moving with its concentration gradient. So since they're moving in the same direction, we call it a symporter. Lastly, based on the information above in your answer to 2A, which molecule can move by secondary active transport? That's going to be our sodium molecule. So bicarb moves with passive transport, so not active transport because it's moving from high to low. However, sodium is being moved from low to high using active transport, specifically secondary active transport. All right, awesome. So last things last, there is one more question on this discussion, and you are being asked to draw and label a graph of secondary active transport of molecule X. Dr. Cheng did not include this in the key because what we did in the previous examples explains it to a T. So don't worry about this one. If you want to kind of see it drawn out, you can refer to figure 5.17D, 6E, and the textbook. Look for the typical saturation graph. But the main thing to be taken away here is secondary active transport is carrier mediated and therefore will always saturate. So it can be saturated because secondary active transport is carrier mediated. Okay, so that's all I have for y'all today. Sorry it took me so long to get this posted. It was a very hard PowerPoint to make and convert from my paper notes. So I apologize for those few errors that we had there, and again, that delay. But hopefully you can watch this a few times and have a better idea of the answers and how to work through problems like this before exam 1. If you have any questions, please attend my office hours or email me to get something scheduled. If you have a very advanced question, please don't put it all in an email because it does take a long time to type up an answer, whereas we can just get on Zoom or something and I can explain. But thank you very much for watching, and I will see y'all later. Bye.